You go through the process and help you.
Okay, you can take your seats. Take your seats, please. Thank you. So let's continue our journey on the secretory pathway. You guys had a good weekend? You studied? Very good. Of course. Of course, because you listened to me. Right? So you've noticed I posted, I'm sorry I was a bit late because I traveled the same day of the last lecture, so that's why there was a delay in posting the recording. But I sensed some tension, some stress, like, is he going to have a recording? Yes, I'm going to have a recording. Okay, so let's pick up where we left off, which was right here. Co-translational translocation. Okay, so just to give you a very quick overview of the stuff we're going to talk about throughout many lectures, translation invariably happens in the cytosol from mRNA themselves exported from the nucleus, right? So that's transcription and so on. A whole bunch of proteins are directly synthesized and then subsequently addressed to their corresponding compartment where they have to work. And we're going to talk about those after we deal with this other main kind of protein trafficking route, which is the secretory pathway. So last week, I gave you a lot of details about what happens when translation is stopped upon emergence of a signal peptide or a signal anchor when it's not at the N-terminus, right? And that's, what does that involve? What's the protein complex that does that? SRP.
SRP.
Come on. Okay, so SRP catch that, brings the whole complex to the ER, interacts with the receptor, helps translocate the protein, and leaves the complex, translation resumes, and co-translational translocation happens, right? So today we're going to continue to talk about what happens in this secretory pathway here, especially once the proteins have made it into the ER or at the ER membrane and are now fated to be transported towards the Golgi complex, and for some of them later on in the secretory pathway, but we're going to talk about those things later. So we talked a lot about this co-translational translocation. Today we're not talking a lot about modifications of proteins. This is going to occupy us in an upcoming lecture. That's the idea that proteins, upon being translocated, they can be modified, right? So I'm just giving you a taste of this. So you can have addition of sugars, modifications of amino acid residues, and so on. So this all happens very early in the ER, also at the same time as the protein is being translocated. So there are a lot of simultaneous events happening. And then depending upon what the protein looks like, it's been transported through the secretory pathway. Right. So this image here, this is transmission electron microscopy. Hopefully all of you, most of you will recognize that. And I really like this picture because, again, you know what that means. Because from a structure function perspective, when you look at the picture carefully, you can actually recognize many pieces that are very meaningful for this secretory pathway, right? So even before I make the annotation up here, you can see here, and it's kind of oriented in some way, starting from this end of the picture all the way to this end, right? So you can already see the organization at the level of the cell. Right, it starts with this compartment. So this compartment, we've seen it before, right? When you look at it, you see kind of clear lumens delimited by electron-dense material. That's a membrane. And on the outside, opposed to it, you have these big, well, these macromolecular complexes. These are the ribosomes. That's exactly right, right? So that's right there, an image, right? If you go talk to your grandmother and she asks you what you study, well, you can show this image and you can explain, well, you see here, this is the membrane, here's the ribosome, and this is what's going on, and you can impress people like that. I guess, I don't know. So anyway, today the whole idea is that proteins have been translocated into the membrane or into the lumen, and now they are on their way to this secretory pathway, right? So then you can see this feature here is very nice, because what you're seeing is that this protrusion in some ways coming out of the ER, but it's devoid of ribosome, and instead, if you look really close, there is some material around it, some electron-dense material. So we're going to see that this is the coat. This is the protein complex that helps form the vesicle. And indeed, once this vesicle breaks off from the ER, you have these vesicles here, and they wind up fusing with the Golgi apparatus. And we're not going to talk about what goes on, about the Golgi itself. We're going to talk about it a bit today. But mostly then proteins mature, the Golgi matures, and then you end up with these secretory vesicles where you have different looking material, and that's this electron dense look also reflects some maturation, some biochemical processes that happens in those vesicles. We're not going to talk much about that today. And finally, this image even shows you this, which looks kind of weird, but that's basically a so-called multivesicular body. So this is evidence for lysosome activity, right? The cell basically digesting stuff. Again, we're not talking in details about that today, but that's what I like about this image. It shows you all these different steps in the secretory pathway. So even though it's a dynamic process, in one snapshot of a cell, you can actually see all the different steps at once, or evidence for all the different steps at once. Okay, so I'm repeating myself on purpose because, again, like I told you last week, this is one way it sticks, okay? It's not, anyway. So we talked about this co-translation and translocation, and today really the idea is how do proteins make it to the Golgi? And one of the key aspects of today's lecture is this process of vesicle formation, transport, and fusion. Right? So I'm going to give you a lot of information about this. But you can already imagine the kinds of needs that this system has to fulfill. Okay, so one of them is what? Is it going to, so these proteins that need to be transported, they are called cargo, right? It's a generic term. So with the cargo, are you going to rely on chains to make sure the cargo gets through? No, right? And we talked about it. The cargo needs to be addressed, so it contains sequences that act as zip code and that the machinery has to recognize. So that's one thing to keep in mind, right? To help you remember the how and why of all this system. Then the next thing is, so you're going to package these cargoes, you're going to package them in a vesicle. I mean, this is not trivial. Okay, so we have to think about, okay, how does the system recognize what to bring into the vesicle? And then how to make the vesicle? Okay? Now, is that going to be enough? No. That was a planted question, right? Why is that going to not be enough? Because the whole point is not just to make a vesicle. It's to make a vesicle that's going to wind up fusing with a specific compartment here, the cis Golgi. Right? So what do you need for this? You need at least two different things. You need also one way to for the vesicle to recognize its target, right? So that vesicle, you don't want to ship it directly to the plasma membrane, right? So the proximity with the cis Golgi helps, but that's not enough. So we're going to see that there are some molecular signatures that help the two actually get together. And then you also need to physically fuse. Again, it is not trivial, although it's helped by the fact that the membranes are essentially hydrophobic and the cytosol is hydrophilic, so these membrane fusion happen. They are thermodynamically favored, if you will. Okay, so we're going to talk quite a bit about that. And then I'm going to get back to that. But the idea, again, giving you the broad general view, is that for many proteins, making it to the cis Golgi is not enough. It's not the end of their journey, if you will. They still have to be further modified. Again, we're going to talk a lot about that in near future. And alongside these modifications of proteins, the compartment itself matures. And so what we're going to see is that there is this so-called cisternal maturation. And the whole idea is that a given cistern, so imagine you have one compartment of Golgi. It starts as cis Golgi, young and immature. But then what happens is that same compartment changes over time. Okay, it's the same, but it changes. It matures. And new cis Golgi are being created. Okay, so gradually, again, we'll see that the cis Golgi cisternae become trans Golgi cisternae, but it's still a unidirectional kind of one-way road. Okay, the content change, and we'll see why. And finally, when these cisternae reach the so-called trans Golgi network, now this is called a network because this is the dispatching platform. So this is where it's more complex structurally, and that's also where different proteins are packaged into different vesicles and sent to different places, right? So some proteins are secreted, so the vesicles are sent to the plasma membrane. They can be different, it can be regulated, it can be constitutive. Others are not sent to the plasma membrane, instead they are sent to other compartments like these endosomes and the lysosomes. Again, this is an overview. We're going to talk in great details about all of these steps, right? Just not in one lecture. Okay, so that's the so-called anterograde movement. Okay, when proteins are trafficked from their place of synthesis and translocation to the outside. Okay, but you need, okay, so I want you to think about this. So what happens if these compartments here, they use their material to ship proteins in the anterograde pathway? I'm going to tell you what happens is that they use their own material, right? So for example, a protein that has to work in the ER, many of them, they get shipped alongside the cargo. Same thing, the cis Golgi proteins, they tend to mature and move along with the rest. So, but that's not where they work, okay? Plus, it's depleting those proteins from the first compartments. So there's an essential need for this so-called retrograde transport, okay? Which is essentially recycling material from the more advanced compartments to the more the more original compartments, if you will, right? So anterograde transport has to come with retrograde transport. So this is absolutely necessary for the homeostasis of the system. Have you guys heard this term before? Yeah, right? Homeostasis is just a steady state maintenance of the whole system. There's constant flux, anterograde and retrograde. So at any given time, the cell looks like it has all these compartments, but they are never exactly the same, right? There's constant flux. Okay. So let's talk a little bit about some tools used to study this secretory pathway. So this one, I'm going to show you some data because we liked it and we used it in my lab. So I actually peppered this lecture with a couple data points from my own lab when I, actually it's a few years ago now. So this is a protein, VSV-GGFP, for vesicular stomatitis virus, G protein. You don't have to worry too much about that exact name. The whole idea is that it's a viral protein, and like many viral proteins, it hijacks the secretory pathway, and it's a glycoprotein, so it's being translocated co-translationally, and it goes along in the secretory pathway. Okay, now the trick is that it's been engineered, so there are two tricks. It's been engineered to fluoresce, so it's fused to green fluorescent protein. You're all familiar with that, right? GFP. Good. So when you use fluorescence microscopy, you can see this GFP, and that's where the VSV-GGFP is. Now the trick is, if you keep your cells expressing this protein at high temperature, 40 degrees, they're not dying, it's like fever, but it's too high for comfort. Okay? So that protein is actually not folded properly. Okay? And there's a phenomenon which I'm going to tell you again in a different lecture, that if proteins are misfolded, they are not allowed to leave the ER. The ER is a place for quality control as well, right? So here it's used as a trick to keep that protein in the ER and accumulate it. So that's what you see here. And then eventually what the researchers do is switch the temperature to a lower temperature of 32. It may be a bit cold, but that's still, you know, comfort. So now the protein can fold and secretion can resume. Okay? So it's basically at a given time point, it's like you give it the start and now it goes. Okay? It's just a trick. And so what you see is first accumulation in the Golgi and then transfer to the plasma membrane. And you can actually watch this live. Right, so you see at the beginning is the ER, so it gives you an idea, right? In spite of all the cartoons, the ER is really kind of everywhere. It's more concentrated around the nucleus. It's actually forming a continuum with the nuclear envelope, the outer membrane of the nucleus. But other than that, the proteins distribute everywhere, and the ER is almost everywhere. And then upon lowering the temperature, things start happening. So you see, you have very quickly, you have vesicles that form, and very quickly they accumulate in this area. That's the Golgi. Then at some point, they start, you have more vesicles. Now they move more in a centrifugal manner. And then you start having, so the plasma membrane localization is less clear because, right, there's plasma membrane everywhere. This is not confocal microscopy. It's kind of no movie at this point. Right, so that's one way where you can really track the trafficking of proteins. If you can time the moment it starts and then watch it go, and you can quantify that. So, and indeed, when people try to quantify these kinds of movies, basically, you can see that very quickly. So at the beginning, right, the protein is accumulated in the ER, and the first thing that happens is the fluorescence, or the abundance in the ER goes down very quickly. So that's an indication of this packaging and sending out of the ER. Okay? The first compartment that gets filled with it is, of course, the Golgi. So right there you have evidence that when proteins leave the ER, where they go first is the Golgi apparatus. Right? So there's a very specific unidirectional flow from ER to Golgi in this case. There's also the retrograde, as we see. And then they have to transition through the Golgi in order to reach the plasma membrane. So that's kind of a more dynamic view of this overview I was giving you, right? ER, Golgi, trans-Golgi, and plasma membrane, and so on. Okay, so if you can visualize the trafficking, now you have a tool, you have a handle where you can start to perturb the system and understand what components are necessary for its function, right? So this is an important point for all of biology. Even if you don't care about this, whatever you do in experimental biology, you need at least these two things. An assay, something to report on what you're studying. So here the positioning of this VSV-GGFP, and a way to perturb the system, to interfere with it, to probe it. Okay? So here again, this is data. Actually, this is data from my lab. Not again, the previous one was not from my lab. So we studied these little embryos, and I'm not going to tell you a lot about them. But here you see a closer view, a confocal cross-section. We're expressing the same VSV-GGFP in the epidermis of the organism. And you see this beautiful accumulation at the plasma membrane. This is an epithelium, right? So cells are tightly adhering to each other, and the protein accumulates at the plasma membrane. Okay? So here, these animals, they hate it at 30 degrees. They want to be at 18, 20. So we can't use this thermosensitivity of the protein. Instead, here what we're doing is we're blocking the secretory pathway in different ways. So for example, and I'm going to tell you about those, you can treat the organisms with a drug, here brefeldin A, which blocks a very early process, and I get back to that, but that's the retrograde transport from the Golgi to the ER. Or you can express a dominant negative version of this protein SAR1 that's also going to block ER to Golgi transport. And when you do that, and you look now at VSV-GGFP, you see now you no longer have this almost exclusive labeling at the plasma membrane. Instead, the protein is everywhere and it particularly accumulates in the nuclear envelope. That's a hallmark of ER accumulation, right? So it's a very clear example of an assay, a protein that's normally trafficked through the secretory pathway, but if you block specific steps, especially in either the anterograde or the retrograde transport, that protein no longer makes it out of the ER, okay? All right, so to be fair, a lot of these details of the secretory pathway, which are arguably universally conserved, right? I showed you the tunicate example, this organism that is close to vertebrates, but the basics are conserved, you know, they exist in yeast. And they were discovered in yeast using these thermosensitive mutants, right? So that's the perturbation. And these kinds of assay looking at proteins which are normally trafficked. And in some mutants, the trafficking is not happening normally. And so you see a different pattern. But the exact difference tells you where the mutant, the mutated gene acts. Let me explain that, right? So again, if you look at a protein which in a normal cell is secreted, you should see it all the way throughout the secretory pathway. But if you use a mutant, for example, this SEC61, you guys remember what SEC61 is? I don't need you to remember that, actually. But that's the translocon, right? That's the very protein required to translocate secreted proteins through the ER membrane. So when in this mutant, what would you expect? That protein does not even make it into the ER. You could even discuss whether it's going to be synthesized at all. But if we assume it's synthesized, and it is to some extent, you have the protein that stays in the cytosol. It's not going to work well there. Okay? And by that same logic, if you have a protein like SAR1, which is required for ER to Golgi transport, the protein accumulates in the ER, and so on and so forth. Okay, so this historically is one way people have identified genes in the yeast required at different steps, right, in the secretory pathway for proteins to be efficiently trafficked, especially in the anterograde, but also in the retrograde way. Indeed, and I'm gonna, that lecture is kind of peppered with mentions of the retrograde transport, and I'm gonna emphasize it wherever it comes. But this, in some cases, the mutant is required, the gene that's mutated is required for this particular retrograde transport. In the 2 slides ago, when I showed you this Brefeldin A treatment, this drug actually blocks the retrograde transport. And that was a neat illustration, because if you remember, in that slide, the protein was stuck in the ER. Okay, so you could be wondering, well, wait a minute, you're only blocking the retrograde transport, so why is it not stuck in the Golgi? Well, because by blocking the retrograde transport very quickly, you actually deplete the ER from proteins that are necessary for the anterograde transport. Okay, so as a result, what the proteins can't even be trafficked from the ER to the Golgi. Okay, so I want this to sink a little bit, because this is a testimony to the importance of the retrograde transport for the homeostasis of the ER. Okay, for even the ability of the system to achieve anterograde transport. Okay, some of you look mesmerized. That's great. Okay, let's move on. So how does that happen, right? I gave you a lot of kind of big pictures. Now we're going to dwell down on some of the nitty-gritty and see how this happens. So one of the first things we saw, and we're going to spend some time looking at, is the formation of these vesicles, right? So I told you when proteins are in the ER compartment, they have to be packaged into these vesicles in order to be shipped forward in the anterograde pathway. So here in this image, you're looking at a very close-up view of what would be an ER part of the ER compartment. So you see this slightly electron-dense membrane, and you see these regions sticking out, literally. And not only do they stick out, but they are also covered with more electron-dense material. And you see a very good correspondence between, the neck here, if you will, of the forming vesicle and the presence of these proteinaceous component. Okay, so again, in one snapshot, that's illustrating this idea that you need this coat complex, it's called, to form the vesicle and shape the vesicle. So we're going to see how this happens. And so here in this cartoon form, so that's representing the fact that you have the vesicle forming. It's still attached through this neck here. And it's forming in part because there are these so-called coat proteins. So they are structural proteins, they associate with the membrane, and their very shape and the fact that they polymerize on the surface of the membrane is what helps creating the vesicle. Okay? We're going to get back to that. But like we said, this is not enough. You have to associate this vesicle formation with the recruitment of the cargo. Okay? So part of this is achieved through, so you have two kinds of cargoes. We saw that last week. Soluble ones that are in the ER lumen, like this guy, but also transmembrane ones, right? So sorry, like this one. So this one would be itself a cargo, but it's a transmembrane, so it does have a cytoplasmic domain. You remember the importance of the topology? Right? In this case, you always want the signal, the information, the zip code, to be on the cytoplasmic side. Okay? Because this is where the codes are. This is where the machinery to recognize the cargo is. Okay, so in this case, it's illustrated here. You associate the transmembrane cargo with the coat protein directly. In this case, it's a soluble luminal cargo, so there's no way it can be in contact with the cytosol. So for those guys, you have always a receptor which helps to connect, if you will, the soluble cargo with the coat. And there are other components we're going to have to talk about, especially these GTP binding protein here, which plays a major role in as a switch to start the formation of this coat. Okay, so just to illustrate a bit further the structural aspect, so this view, I have it there for illustration and also because it's kind of cool. It's actually a scanning electron micrograph, as if you were underneath the plasma membrane, and that's endocytosis. So it's not ER to Golgi, but it's the same concept, right? You have vesicles forming, and they form because of a coat of protein surrounding them. And in this case, these are so-called Clathrin pits. We're going to get back to those over and over again. And you see it's highly structured. Okay? So this is to illustrate the fact that you have different kinds of vesicles that are formed using many different kinds of coat proteins. And I might want you to remember the main kinds of vesicles and coats, but not the details of what makes up the coat. So for example, this one here, ER to cis Golgi, there's a dedicated coat machinery for this. Okay? It's only acting in the ER to cis Golgi. It involves these small GTPAs, SAR1, remember SAR1, the dominant negative blocks anterograde transport. And it uses all these proteins in the coat, and overall it's called COP2. Okay? The cis Golgi to ER, so that's the retrograde transport. It uses slightly different proteins. It uses a different GTPAs. That's called COP1. So here you see, just by the very nature of the coat, already carries some specificity. Okay, so the proteins that will be exclusively involved in coating vesicles for Golgi, ER to Golgi transport is COP2, Golgi to ER, COP1. So you have some specificity, and those guys that's all they do, right? The other kinds of trafficking, which we're not going to talk much about today, they involve clathrin and other kinds of adapters and code proteins and a different GTPAs. Okay, so there is a lot of specificity built in the system, but the general rules I'm going to tell you about today can apply throughout, because if you look at this table, what it illustrates is, although there are different kinds with specific transports, there's always a GTPase and there are always code proteins. Okay, so let's see how this works. So first off, very briefly, again about the code proteins. In the case of the clathrin pit, so clathrin has been perhaps, very well studied. And here this cartoon illustrates the relationship between the structure of clathrin and the formation of a vesicle. In the lecture on endocytosis, I will show you even more about this. But what I want to really emphasize here is that, so this is a complex multimeric protein. You have these heavy chains here in red and these light chains, and they form, so right here you have already 6 proteins. And it's forming a so-called triskelion here. So here one triskelion is illustrated. And what I like about this is that, so the triskelion, although here it's represented flat, it's actually a 3D structure. So that's not going to be on the recording, but if you look at my fingers here, and there's stuff that points inwards, right? So it's slightly bent. So I only have 2 hands, unfortunately, but if I had enough, I could build an entire vesicle just like this, okay? Just by virtue of the triskelion being slightly bent, okay? And you have essentially it achieves 2 things, at least. One of them is through these sites here, which are pointing internally, it helps bind with specific components towards the membrane side, okay? But then the shape itself, when these triskelion further multimerize, they start making this vesicle shape, okay? So you have a direct relationship between the structure of the proteins, the multimeric complex, and the vesicle forming. Okay, now, one of the key questions is you don't want these code proteins to start assembling randomly anywhere, right? You want them to assemble at the membrane, at the ER membrane, at the Golgi membrane, and so on. And ideally, you want those assemblies to be coupled with recruitment of the cargoes, right? That's one of the key concepts here. And one of the main switches for this are the small GTP binding proteins or small GTPases. For example, the SAR1 or ARFs. So this is a general family called ARFs. Now this cartoon here looks really like nothing. But have you guys heard about the small GTPases before in principles? Not? Okay. So let me drive this point home right away. This principle, the small GTPases, this is extremely important. We're going to see that over and over again in almost every lecture. If I have to choose one concept I want you to walk away with in this entire course, this would be it. The idea that you have proteins that can bind GTP, and when they are bound to GTP, they are active because they are in a particular conformation. That's the active form. Okay? But then they can also hydrolyze GTP. They're just really bad at hydrolyzing GTP. Yes?
Is it similar to like the G couples or something?
Not exactly. I mean, it also uses GTP or GTP binding proteins, but those are much more common than the G-couple receptors is slightly different principle. That's a good question. So the point is, when they hydrolyze GTP, they become GDP bound. They don't spontaneously let go of GDP, and that's an inactive version. So this system, which we'll see is everywhere, acts like a switch. Okay, the protein is either GTP bound or GDP bound, active or inactive. And the transition from one state to another is always catalyzed by a GAF, guanine exchange factor, or a GAP, GTPase activating protein. So it activates the GTPase function of this ARF here. And in other words, so GAP will be an inhibitor. Okay? Now, I know that I'm emphasizing this because you have to think about it a little bit. It activates the GTPase function, but that causes a change of state, going from GTP-bound, the active form, to the GDP-bound, the inactive form. So that's why it's an inhibitor. Okay? And the GAF, and what we'll see is how the GAF activates. Sorry. So the GAF is an activator because it catalyzes replacement of GDP by GTP. Okay? So the function of these proteins is not because they can hydrolyze GTP. Okay, that's the confusion most people always have. It's not what makes them functional. They are not metabolic enzymes. Okay, it's just a conformational switch. So let me illustrate this in this case. In the example of SOR1. So most SOR1 by default is inactive. That means GDP bound. Okay, and if there's again, that's something you might as well start learning right now. GDP bound inactive. Okay? To activate it, you need a GEF. And what the GEF does is foster replacement of GDP by GTP. And so here the GEF happens to be this protein SEC12, right? SEC because it was identified in these mutant screens in the yeast involved in the secretory pathway. So it's at the ER membrane and it catalyzes recruitment of now this active SOR1. So just to illustrate how GEFs work, this is another kind of small GTPase of the RAS. So that's RAS itself. So of course I do not need you to remember all the details here of the structure. But so the idea is that you start with an inactive form bound to GDP, and GDP is here in this pocket of the protein. What the GEF does, right? So guanosine or guanine nucleotide exchange factor, an activator. What it does is simply coming and kick out GDP, right? So there's this alpha helix. For RAS, the GEF is called SOS. Doesn't matter at this point. We'll see that again, or you'll see that when we discuss the FGF MAP kinase pathway. But the whole point is it comes in with this alpha helix, kicks out GDP. And spontaneously, this leads to the replacement or the addition of GTP just because in a cell, GTP concentration is orders of magnitude higher than GDP concentration. Okay, so by default, if you kick out GDP, now GTP comes in place. And the whole idea is that GDP bound versus GTP bound, the conformation is not exactly the same, especially on this switch one and so on. Again, I don't need you to know the details, but just remember this very basic concept that this is a conformational change, right? So in cartoon form, you have this one, it's inactive because it doesn't have this little thing sticking out, right? The activity is not due to GTP hydrolysis. GTP hydrolysis instead inactivates these kinds of proteins. Okay, so back to the ER. Activation of SOR1 causes a conformational change. We just saw that, which exposes the C-terminus that's hydrophobic and can now be tethered to the plasma, to the ER membrane. So now you have this protein at the membrane. So what do you think it's going to do next? Start recruiting the coat. Okay? So indeed, the presence of active SOR1 GEFs here starts recruiting the code and basically triggers the polymerization of these code proteins. Okay, so that's the key switch. And of course, very importantly, the first subunits to be recruited interact directly with SOR1 here. That's this SEC23. And it often comes with SEC24. Again, secretory mutants. They form dimers, but SEC24's specialty is not to interact with SAW1, that's just the tether, it's to interact with the cargo. And there you go. So this is, in a way, this model here is really beautiful in the sense that it conveys very important principles, right? How do you assemble at the plasma membrane the main components you need, which is the coat to make the vesicle and the cargo here, and how this is triggered not randomly, but by activation of the small GTPase SAW1, right? And you'd say, well, you can now walk away and the lecture is done, but that's not the case. We still have a lot to talk about. Okay? But that's a very important step. Activate SAW1, start recruiting the coat, start recruiting the cargo. And you nucleate essentially what you need to do to make these vesicles. Okay, so indeed this nucleates because then there's kind of a spontaneous polymerization and recruitment of more cargo, and there's usually more than one SAW and so on. So you're making a vesicle. Right, so at this point I consider, okay, we know how we make a vesicle by now. Okay, and eventually the polymerization of the coat surrounds the entire vesicle, narrowing the neck, and then when you have two hydrophobic membrane coming very close together under the pressure, if you will, of a polymerizing coat, it tends to break off, right? And so there's a step in which the vesicle breaks off from the ER membrane and the polymerization of the coat is completed, right? Completely surrounding the vesicle. So now you have a free vesicle, okay? Is that enough? No, it's not enough. Because I'm just gonna tell you right away, if you leave the vesicle like this, coated, it can't go anywhere. It's not gonna be able to find and fuse to its target compartment. So what happens next? What happens next is, here is illustrated, well, begins to be illustrated here, which is if you treat cells with this non-hydrolyzable GTP analog. So what is that? That's a GTP that can be loaded onto SAW1, but SAW1 won't be able to hydrolyze it. Okay? So if you will, it will be stuck in a GTP-bound active form. What does that mean? It means its little tail is going to stick out and it's going to remain at the membrane and is going to continue to hold on to the coat. But so the idea is that, and this was showed briefly in the previous slide, it's right here. Once the vesicle is completely formed, the coat is complete, somehow this acts as gap, right? So GTP hydrolysis by SAW1 happens, right? So that's why you have these little inorganic phosphate. So PI stands for inorganic phosphate. So GTP hydrolysis happens. These SAW1s are now GDP bound, so that's the inactive form. They switch back to folding their little hydrophobic tail back into the protein and they detach. You remove SAW1 from the membrane, you dissociate the coat, okay? See a lot of yawning. Is that boring? Should I spice things up? Yeah? Okay. Fasten your seatbelts. Okay, so, but this is a very important step, right? Because the whole idea is that now this vesicle has been created and it has to be able to fuse to a target membrane. So that's what we're going to focus on now, with two ideas in mind. You have to recognize the target membrane, so where does the specificity come from? Because now the coat is gone. You can't use the coat anymore for specificity, it's gone. Plus, you also have to overcome whatever thermodynamic barrier there may be to fuse with the target membrane. So let's see about that. Okay. Whatever. So that's just a quick rundown of what we just talked about, right? Recruitment of SAW1 or an ARF in other systems. It's always kind of the same principle. You recruit the GTPase, you start assembling the coat, it helps form the vesicle. When you're done, the vesicle detaches from the membrane of origin with the cargo in it, And the coat hydrolysis of GTP switches the ORF GTPase back into an inactive form. The coat deassembles. Now you have an uncoated vesicle. And if you look back at that first image, you see those, right? You can look carefully and you'll see the vesicles close to the ER, they have these electron dense material around them, and further away they don't. So they are ready for fusion. So just a few words about the sorting signal. If they are soluble, I said that before, they need a transmembrane receptor which will help them connect with the coat. But if they are, okay, so here they are. I'm not in love with these tables, so I'm just going to go through them more or less quickly. But again, this is where the specificity of addressing is, right? This is what tells the system that particular protein has to go to that particular location. So you have, there are a few examples we're going to talk more about, such as, well, these ones here. So among luminol sorting signals, the two different kinds we're going to talk a bit more about. So this KDEL, I'm going to have a slide at the end of this lecture. That's for ER resident soluble protein. How are they being trafficked back to the ER instead of being trafficked along the anterograde pathway? And then the mannose-6-phosphate, which we're not going to talk about today, but these labeled proteins that are usually destined to go outside to be secreted or even go to the lysosome. And I'm not going to tell you a great deal about all these other examples. What I want to emphasize, so you don't need to remember this, but the general idea is that, so they're called cytoplasmic sorting signals because they are in transmembrane proteins, so they are directly facing the cytosol, right? So that's what's recognized by the coat proteins. And there's a great deal of specificity, so, well, specificity for the pathway, but then there is some flexibility in the exact sequence, right? It's never one and only one type of sequence. It's more like a consensus, okay? But there's enough specificity in these different consensus to recruit, you know, either the COP1 for these ER resident membrane proteins to come back to the ER, or if I take another example, which I like, this one, for example, the LDL receptor helps recruit this complex with clathrin to go all the way to the plasma membrane, right? So you have this specificity encoded at this level. Okay, but let's talk about what happens next, right? So again, we left it where there's an uncoated vesicle, and now what it has to do to carry along in the anterograde pathway is to fuse with the target membrane. So how does that happen? So one of the first thing is specificity yet again. So there's, you know, how does a given vesicle know, in quotes, to interact with a given target membrane? So this is largely encoded by another family of small GTPases. Same principle, GTP-bound active, GDP-bound inactive, okay? So those, they are active in vesicles, and the RAB, so this is the RAB family of small GTPA. So they tend to be loaded onto particular vesicles, basically marking these vesicles with a destination, okay? And we'll see that the so-called snare proteins, so they are the ones which will help the system of, you know, achieve actual fusion between the vesicle and the target membrane. And we'll see how. So you have the two components, the address, the specificity, and the physical interaction and the physical fusion of the target, of the vesicle with its target. Right, so RABs, again, so this is some data from my lab. Now I wonder why I'm showing you so many of those, but oops, I'm still going to show you. So these are two cells we like to work on, and here what you're looking at is just a 3D projection of, so the two cells, the nuclei in red, and in green, these dots, we can do much better now, actually. They are GFP Rab4, so it's the same principle. You fuse GFP to one protein of interest, and it usually goes wherever that protein normally goes. So what you're seeing here is one of the Rabs. There are many Rabs. It's Rab4, and you see it labels all kinds of little particles in the cell. So those are individual vesicles, and the presence of Rab4 on their surface tags them with information as to where it's going to go. And I can tell you Rab4 tends to label vesicles that have been endocytosed, but are somehow fated to be recycled back to the plasma membrane, as opposed to, for example, the lysosome for degradation. We're going to talk about that more later. All right, so the Rabs, they convey information about the target, and indeed, that's because in the active form, they can interact with an effector, right? So this is another general principle of all of these small GTPases is when they are active, their specific conformation allows them to engage in specific interaction with the so-called effector protein. Okay, that's how they work, right? For SAR1, we saw the effector is the SEC23, recruits it. Here, it's the same logic, sorry. The active form of Rab helps bind to an effector, but the effector, the beauty of the system is the effector is on the target membrane, right? So right there you have the mechanism whereby the vesicle is going to be able to recognize the target where it needs to bind, to fuse. Okay? And then the second step is going to be actual fusion. And so for that, I told you we have the snare proteins of two different flavors. Sorry, let me just pause for a second. You have some on the vesicle, and they are called V-snare for vesicle snare. Okay? And then on the target membrane, you have so-called T-snare for target snare. Okay? So now all you need is to have a mechanism whereby these guys will come together very tightly. So when people studied that, so here you see a structural model of a snare complex assembled. So that means there is both V snare and T snare together. And it so happens that these proteins are so-called coil domain proteins. So they tend to form these heteromeltimers here, which are very stable. They adhere very tightly, okay, because there are a lot of hydrogen bonds and basically so-called weak interactions between these different chains, right? So that really helps, right? The Rab system help dock the protein, the vesicle on the target membrane initially, but then the snare system basically tethers them very closely, okay? And that, so that the stability of this association and the proximity together with some thermal fluctuation will help the fusion itself, okay? Indeed, upon assembly of the snare complex, these two membranes get really close, and that leads to membrane fusion. And now you can see on this cartoon here, the topology remains the same. In other words, what was exposed on the cytosolic side here remains on the cytosolic side here, etc. So that's why I was telling you last week, the topology that's established upon translocation initially matters a lot because it remains the same throughout the secretory pathway. Okay? Now, here you already see one conundrum. How are you going to recycle this? Right? That's that complex here. I told you it's very stable. Right? Hard to unwind. But do you think you need to recycle this? Yes. What proteins do you need to recycle? Yes, snare is not there. Which one, the V or the T? The V snare. You need to recycle the V snare, right? Because the V snare is needed on the vesicle. So you need the V snare when you package the vesicle in the first place. Right? That's got to be part of the package. And, oh, sorry. Yeah, if you block that, so that's also data from my lab. If you block that with a dominant negative inhibitor of the diffusion, you also block the secretory pathway eventually. Okay, so but let's get back to that. So the cis-snare complex is very stable, and yet has to be unwound in order for the V-snare to be recycled back to the ER in this case. And that's the job of this so-called NSF complex. So it has a busy name. Quite frankly, I don't need you to remember this name. It's easy to find. You can remember NSF like National Science Foundation. But you don't even have to, quite frankly. What I want you to get from this is the general principle that this is a very stable complex, the cis-snare complex, that has to be unwound, and that needs energy. So this NSF complex, what it does, it uses the energy of ATP to deassociate the cis-snare complex, right? And the reason why I say that is because this is something, if you think about it, it makes a lot of sense, and you can understand that, right? It's more conceptual than just the details of what NSF stands for. Don't tell me it's National Science Foundation. That was a joke. Okay, so anyway, it was not a great joke. It was just kind of a lame joke. But so now the complex is deassembled and the V-snare can be recycled. How is that going to happen? Well, they're going to be packaged in one of those recycling vesicles using the COP1 system and back to the ER. Simple. Okay, indeed. So this is just a quick recap. Yeah, so that's the point I was making, right? There is some specificity between the anterograde transport and the retrograde transport COP2 codes are involved. So this is that's the only step that requires SAR1, and all these COP2 protein is specialized for ER to cis-Golgi, and vice versa. You have cis-Golgi to ER that uses COP1, different GTPAs and different codes, right? So these V-snares and a couple other proteins, they can be recycled now through this pathway. Okay, so we're going to talk a little bit about that. How are we doing with time? We have another 15 minutes. Oh, so I could take my sweet time. I thought I was late. Okay, so again, I'm just going to give you a brief recap of everything we've talked about and kept emphasizing the need for recycling. Right, so we started with making this code here, SAR1 GDP is activated. So here I say signal question mark. What do you think, if you had to make a guess, what do you think could be a signal to activate SAR1, to tell it, you got to start making vesicles here? Just a wild guess. Just think about the system.
The opposite of gap.
Yeah, well, that would be the mechanism, the gap. Mm-hmm. But why? What would I? Because usually gaps themselves, they have to be activated, right? It's not part of the lecture. I just want you to try to think about this. Well, that would be, is there any point in making vesicles if there's nothing to transport? No. So you could imagine that an excess of cargo, or an abundance of cargo in general in the ER or at the ER membrane might help trigger this first step. Okay. So first, activation of SAR1 GDP, recruitment of all of these cargo proteins with SEC23, SEC24 initially, and that's a key step, right? Because it associates the active ARF GTPase, SAR1, with the cargo and the coat, the key components you need to start making that vesicle. Then you polymerize the vesicle further until, so you don't need to remember all these steps. And now you have a COP2 coated vesicle. We saw that the complete vesicle causes hydrolysis of GTP, and now you have an uncoated vesicle which is ready to fuse with the cis Golgi. And that specificity, what protein encodes that? A RAP protein, right? Associates with an effector on the cis Golgi, and the snare complex can form, helping with the fusion. NSF unwinds the snare complex. These snares now have to be sorted back. So I'm going to finish with this a bit in advance. I'm sure you won't hate me for this. Like you see, this snare protein is here, and right, it has to be here. That's a transmembrane protein. If I were to ask you, how do you think this transmembrane protein gets there in the first place? Translation of translocation. Yeah. Right? Yeah. Last week. Yeah. That's how it gets there. Right? So that's the only source initially, and then it has to be recycled. Okay? So that's how the V snare gets there. It has the signals to be incorporated in these vesicles to interact with the coat and so on. So it works mostly here, and then it gets the C snare complex gets deassembled, and the V snare, specifically the V snare. You don't want to package the T snare in the COP1 vesicle, okay? Just the V snare. And some other resident proteins, so they are packaged in this COP1 protein and back to the ER. Now you have another kind of snare complex. So you can imagine that some of these snare proteins, right? So they will shuttle back and forth between the two compartments. Okay? Many of the components needed for this transport, they will just shuttle. That's the flux, the constant flux, this homeostasis of the ER and cis Golgi. Okay, to finish, I'm gonna show you, and actually, I hope you still have some energy, because the last slide, and it has a very elegant mechanism I am particularly fond of. So that's the mechanism to recycle resident ER proteins, but the soluble ones. So not the ones like the snare that have a transmembrane domain and a cytoplasmic region with a signal. The ones that are soluble. And the soluble ER proteins, so some of them could be, for example, the chaperones we talked about, or we will talk about more. So there are a few resident ER protein, also glycosyl transferases and so on. There are quite a few of them. So they have to work to be in the ER, but somehow they randomly get incorporated into vesicles. So there's a mechanism to bring them back. Okay? And usually, so they all have this so-called KDEL tag. But again, themselves, they are soluble, so they cannot interact with the coat, especially the COP1 coat for retrograde transport. So there's a receptor, KDEL receptor, which itself is always also packaged in these vesicles. Right? So these vesicles have a lot of things going on. So the KDEL receptor, and as you can see here, the protein is not interacting with the receptor. And it makes sense because there's no need for this protein to be shipped to the Golgi. It's an ER resident protein. But the receptor is. Now what happens is on the cis Golgi, now there is an interaction, right? So now that packaging of the ER resident protein is specific. It's specific and it's mediated by interaction with the KDEL receptor. So now that's all the system needs to bring back these proteins to the ER. And interestingly, once they are back in the ER, so the receptor is not depicted here, but the protein and the receptor deassociate. So it's kind of weird. You have some association where it's needed, no association where it's not needed. And the elegant aspect of this is something we're going to see over and over again because it uses a feature of the entire secretory pathway, which is a pH gradient. Right? So the idea is that you have slightly higher pH here. No, sorry. The other way around. Lower concentration, so higher pH in the ER compared to the more advanced vesicles. So that's a general rule. Okay? Lower pH, more acidic in the more advanced compartments. And the simple trick here is that the interaction between the K-del peptide and the receptor is pH dependent. At higher pH, lower acidity, the interaction does not happen. That's the ER situation. And that's essentially the situation in the vesicle. But at slightly lower pH, so higher concentration of protons, in the cis Golgi, it's subtle, right? But it's enough to now have a specific association between the K-del peptide and the receptor. So although the receptor keeps cycling, right, because you need it in the cis Golgi, so you have to send it there all the time. And it comes back. And you need it both ways, actually. So the receptor is cycling, but the ER resident protein, on average, they only are recycled, right? Because they do not associate specifically with the receptor in the anterograde pathway, only in the retrograde pathway. And that's all due to a difference in pH. And we're going to see in future lectures that these differences in pH, they regulate the protein-protein interactions in specific compartments at other places in the secretory pathway. All right, and I'm going to stop here.